Well, welcome all this huge audience that is in the room, and welcome to all of you who are online. We are starting the last section of our soil science seminar series today. I'm going to take a couple of minutes just to remind everybody about the organization for the next 3 weeks. Today, we're going to hear from 3 speakers in a format that's similar to what you might find at a professional meeting. So each of the speakers will have 12 minutes, and then after each talk, we'll take about 2 minutes for questions. And then we'll go to the next speaker. So that's our seminar today is actually going to be 3 seminars by the students who are in the course around 600 feet. Next week, we'll have something similar. We'll have 1 12-minute talk. That will be followed by 4 5-minute talks, which will be in the rapid lightning format. In that case, with the lightning talks, we'll kind of go through them lickety split. Well, okay, not super fast, but anyway, without pausing a whole lot. And at the end, it will take time for the students to respond to questions over what has been. In the final week, we're going to try something really different. That's where the speakers will each have 8 minutes to speak to the audience without visual aids. And the idea is to present a talk that will be reasonably interesting and comprehensible to a non-scientific audience. So that's what's going to be on the front end. So that's the plan for the next 3 weeks. Today we have 3 speakers, and I'll introduce each one as they begin. And so today our first speaker is Jessica Nelson. Jessica is a native of Virginia, but she's been in Iowa for a while. She came to Iowa to go to college and obtained a bachelor's degree in sustainable living with an emphasis on living systems and living soils, which is a wonderful name for a major at Maharishi International University. That was in 2013. Then she went to the United Kingdom and got a master's degree in soil science at the University of Aberdeen. And now she's working on a PhD here at Iowa State in soil science and environmental science. She's working with Marshall McDaniel. So the title of Jessica's talk is Miscanthus by Giganteus Changes Soil Structure and Soil Water Balance.
Thank you, Dr. Thompson. So, yep, that's what my talk's about. I'm really interested in how soil structure changes and soil water holding capacity changes due to the crop that's being grown on it. I used data that was collected by Jacob, Studt, and Perla Carmenate, as well as had help from Maoz for X-ray CT scanning. Sorry, I did it. Cool. So I just want to start by giving a little bit of background on soil architecture. It refers to the arrangement of the soil particles and the way that they're being held together, forming aggregates, pores, and channels. And then this spatial arrangement determines the soil structure, which affects its physical, chemical, and biological properties. Here is a little diagram about the soil architecture. You can see all of the little brown aggregates here. You can see different films of water in the pores. You can see the large macro pores. You can see some roots exploring that, some fungal hyphae exploring the aggregates themselves, some bacterial colonies hiding in there, and organic matter. And you can see a whole ecosystem happening to help create this structure whether that's different environmental factors such as wetting and drying and physical things that are forming aggregates, or biological or chemical. So pore space and water, what is that? What's the correlation there? So the size of the pore space in the soil will affect how water flows and is held within it. So we have those larger pores I showed you, those macropores. They allow the water to flow more freely through the soil. Then you have the smaller ones, which were the mesopores and micropores that help hold water more tightly, which influences the availability of it to plants and how hard or easy they have to work in order to access it. And then the size and distribution of the pores in the soil can influence the soil's water holding capacity, drainage, and then also aeration as a result, and then in turn affect the plant's growth and nutrient cycling. So my research overview is that I investigate how different crops affect soil structure and porosity and how this influences soil water holding capacity. For my experiments, I did some sites in Iowa. They are known as the LAMP sites, the long-term assessment of Miscanthus productivity and sustainability. You can see here, I used soil aggregates from two sites, the central site and the northwest site. These sites were similar in pH, total carbon, soil texture, rainfall, and temperature. And the crops grown at both of these sites were Miscanthus giganteus and a no-till maize. And the Miscanthus giganteus is really a rhizomaceous perennial biomass crop, and the annual maize grown is also produced for biofuel. I have a few hypotheses that I'm going to walk through for the research, and the first one is that the soil aggregates from soil around Miscanthus, I think that they'll have more pore space, and then as a result, I think that this higher pore space will increase water holding capacity as compared to maize. So the experimental design for this is that I measured total porosity of aggregates from both Miscanthus and maize at the two sites using X-ray CT scanning and image analysis, which you can see here. Is basically we took aggregates and put them in a straw, put it into an X-ray CT scanning machine so that we could get the slices and then stack them up in order to form these really pretty pictures of the pore spaces, which I'll show in a moment, a little bit closer up. The results of this experiment were that the total porosity within the aggregates, and granted this is total porosity above 5 microns because that's the resolution of the machine. So anything below 5 microns, and we don't have that recorded in here. So these are this is total porosity above 5 microns. And at the northwest site, there was not really a significant difference between the porosity of the aggregates. And at the central site, there was a difference. The Miscanthus had more of that total porosity. And these are 2 to 8 millimeter aggregates that are being looked at. Here's images of those 3D renderings I told you about. Here's the aggregates at the top. And then when we remove the solid soil and filled in the pore space, then you can now see that in 3D and kind of see the distribution of those pores. The brighter the color, the larger the pore. This is not quantitative, this is more qualitative, just so we can see what's happening there. My second hypothesis was that the soil around the Miscanthus was going to have a higher water holding capacity than maize. So we looked at the porosity, now we're looking at the water holding capacity. And what we did was set up a standard procedure for water holding capacity to measure it of the aggregates. And our results were that the Miscanthus held anywhere from 12 to 15%, about 12 to 15% more water in its soil than the maize did. And this is the northwest site and the central site. So that was interesting to see that happening at both locations. And then the third hypothesis was that when we take these samples that we looked at and we destroy them, and then we measure the water holding capacity, how will that destruction of the structure impact the water holding capacity? So we think that the intact aggregates will hold more water than the destroyed aggregates. Because you can see here, they're going to be more tightly, the pore space is going to be reduced overall. So we set up the experimental design and we took the aggregates, and I'm calling them intact aggregates, which is just whole aggregates, and then they were ground up in a mortar and pestle to destroy the structure. And we did do particle size analysis to make sure we didn't alter the texture during that process. And so this is what it looked like before when it was intact, and then when it was destroyed after going through the mortar and pestle. And the results from that are that for most of the soils, the water holding capacity decreased when you destroyed the structure. So here's the intact Miscanthus, and then here's the destroyed Miscanthus, and you can see this difference here is the water held due to aggregate pore space is what we're assuming. And it held true for most of the sites except for the northwest maze. It seemed to increase slightly, and we think that could be due to one thing being that there weren't enough replicates to really give us confidence in it. It's like that could be, if we did more testing, maybe we could get a better trend. Or it could be that a little bit of homogenization happening where you destroyed those finer particles or the structure, and then those finer particle sizes had more surface area exposure, which could be holding onto more of that water in that specific scenario. So in conclusion, Miscanthus giganteus may be altering the structure and pore space within and around soil aggregates. That increases the water holding capacity of the soil compared to the aggregates from the soil around maize plants. And then the structure and pore space in and between aggregates does influence the ability of the soil to hold water. So I just want to acknowledge all the awesome people that make this possible for me: Marshall, Andy, Perla, Jacob, Maoz, and the ISU Biomass and Nathaniel Labs, and then the Oregon State University for spanning these for me, and then my funding body of CAPI. So I'm ready to take questions.
Thank you very much. Okay, well, let's open up to the folks in our in-person audience. Have a question.
So I have a question about analysis of differences. And I know it's always an educating when people ask about it. You know, the difference was significant. I'm not too worried about that, but for some of your charts, you had like 80 degrees, some not. I like your thoughts where you didn't have letters, did they not pass a significance test, and how comfortable are you?
Yes, this is more in the preliminary stages of analysis. So these are preliminary findings. So I haven't run them through the statistical analysis yet.
Okay, so we need to do the, all right. Well, okay, so if they didn't have letters, they just haven't been found.
Yes. We do have the standard error bars, but haven't run the actual statistics.
So so for some of the conclusions that you made about this was a change, if it doesn't say there's a significant difference.
Right. I may change them. I'm sorry. I haven't been repeating the question. Yes, it's okay. So the question was if the results from the statistical analysis prove that some of the error bars and some of the graphs show that there wasn't a statistical significance, then would I change my conclusion? And the answer would be yes, that's very possible at this stage. I'm still in the analysis process, so I could very well change them. Right now, this is more preliminary study to go into trying to start like doing my next level to dive deeper into exploring those things. Thank you.
What's the texture of the plus ones versus the people?
The northwest. So what's the difference between the texture of the northwest and the central site? Here is a table of the site characteristics. They were both clay loams.
Can you hear me, Jessica?
Yes, I can hear you on my computer. I'm going to have to repeat what you say though, but that's fine.
Okay. You showed that you had some fraction of the porosity measured with X-rays. And you showed some kind of water retention for aggregated soil and soil with aggregates disturbed. And I wonder if you measured the bulk density of any of the aggregates, or if you measured the bulk density or controlled the bulk density when you looked at the water holding of the aggregated soil versus the disturbed soil, whether they're the same bulk densities or different bulk densities.
Got it. Yep, so Dr. Horton was asking, he saw that I was measuring the fraction of the porosity from the X-ray CT scanning and was wondering for when I was measuring the, if I measured the bulk density of the aggregates themselves, not the aggregates themselves, no, but for the water holding capacity measurements, we didn't have a way of controlling the bulk density of those, but the process itself was the same for when we measure out the samples, we put them into centrifuge tubes, and then we line up all of our stations. And so when we add the soil to do the testing, we just dump it in, and that's how we do it for everything. So that is, so there wasn't any other way of controlling the bulk density of that. So
You might
loose soil.
You might want to go back just to, do a side check.
Okay.
And see if the aggregated soil, how you dump it in, and the destroyed aggregated soil, if they pack to the same bulk density or not.
That could help, yeah.
That'll be a big factor on the water holding, I believe.
Awesome, thank you.
Okay, while our speakers are getting set up for the next presentation, let me do the introduction of the second speaker. Our second speaker is Reid Barker. Reid grew up in Ames, Iowa, but he went to South Dakota State to get a bachelor's degree. His bachelor's degree was in agronomy in 2021. Then he returned to Ames to Iowa State to work on a master's degree in soil science and environmental science. He's advised by both Matt Helmers and Marshall McDaniel. And the title of Reid's presentation is Soil Phosphorus Stratification in Long-Term No-Tillage and Cover Crops.
All right. Thank you, Dr. Thompson. Like you mentioned, today I'm going to be talking to you about some of the details and important concepts regarding my thesis. A couple things I'd like to talk about first before I get into some of the details is in the state of Iowa over the course of the last decade or so, the prevalence of no-till and cover crop acres is generally increasing. And if you kind of ignore Illinois and Indiana, you can see that in this figure between 2012 and 2017, that generally the rate of no-till is increasing across the vast majority of the state, as you can see in the dark blue and the light blue colors for each county. And the exact same thing is happening for cover crops on a much smaller scale, but between those dates, the exact same thing is happening. But even so, more for Indiana and Illinois as well. So that kind of gets into a potential issue with that, regardless of the benefits that we get. So like increased organic matter, reduced soil erosion, and better nutrient cycling. A potential issue with that is the stratification of soil phosphorus. And essentially what stratification or phosphorus stratification is it's the net upward movement of soil test phosphorus and other phosphorus forms from deeper into the soil profile towards the soil surface. So every year when you're growing a crop and you're not tilling it under, you're essentially pulling that phosphorus from deeper out of the profile, and then as your crop residues are left on the surface to mineralize or your broadcasted fertilizers are left unincorporated, then they're going to remain towards the soil surface due to how sticky phosphorus is and how little it moves throughout the year. And as you can see here, this is the diagram from Rollins and Voss, which is a really pertinent stratification paper, which kind of just shows you how soil tests can become higher and higher towards the surface and kind of like disparity with depths. And so some of the potential issues of phosphorus stratification is a handful of studies have indicated that you'll have increased runoff losses due to having a higher proportion of different phosphorus forms sitting on the soil surface. That during the rain event, you'll just have more phosphorus in the dissolved form of orthophosphates that can just be picked up by water like nitrates and just lost in that form. And given the texture, you might see more subsurface loss and tile drains as well. So if it's like a really coarse soil, then if infiltration is high enough during rain events, you might also see more orthophosphate losses as well. Another potential issue is poor accessibility of phosphorus in dry conditions. This is not as well supported, but there are a handful of studies that have indicated this to be the case, especially in dry conditions. And the reason for that being is in typically good moisture conditions, roots don't have a hard time moving through the soil. So kind of imagine like you have to grab something off the top shelf, like say a box of crackers or whatever. And the difference is whether or not you have a step stool or not. So in dry conditions, roots might not have the ability to move through soil as well as it normally would. And in a stratified situation, you might end up having more phosphorus up here. And if the roots can't get up there, then even though you might have adequate soil test phosphorus, even high soil test phosphorus, then plants could still develop deficiencies given how deep the root structure is. And another potential issue would be that due to the stratification effect, given how you test your soils, you might end up obscuring your soil tests. So if you take a shallower core, then you might end up getting much more phosphorus than you would if you took a much deeper core. So say for instance, if you took those shallower cores, you'll be capturing all the phosphorus from these top 2 or top 3 layers, just kind of showing this diagram to show how phosphorus levels can change with depth. As you can overestimate your phosphorus test levels if you don't sample deep enough. And so the objectives of my study were at one of our sites to quantify the effects of long-established no-tillage and cover crop treatments on phosphorus stratification at the ag drainage well site in Gilmore City, which we call ADW, and I will from this point on call it ADW. And in addition, to illustrate the potential crop nutrition effects of P stratification, given if it's observed at this site. And so getting into my methods, like I mentioned, the Ag Drainage Well Water Research and Demonstration Site in Gilmore City has been a long-standing site that's been implemented to understand tile drainage flow and the impact of land management. And so the treatments for this study were 32 plots with a 2x2 factorial treatment. And if you don't understand what I mean by that, then it's just like having 2 types of treatments that are combined together to create multiple treatments. And so what they are is having a chisel plow with a winter fallow, and then having a chisel plow with a winter rye cover crop, which is broadcasted by hand, which is kind of meant to simulate like aerial seeding. And then the same thing with no-till, having winter fallow and a rye cover crop. And these treatments were established in 2010. So at the point of my sampling about a year ago, these plots would be 12 years old. And so for those of you who don't understand what I mean by chisel plot, is typically when you think what most people think of tillage, they think of either the moldboard plow or a disc. And what chisel plow is essentially you have an implement that will rip a furrow through the soil instead of inverting the soil. So you can kind of help with aeration without necessarily flipping it all the way over and mixing it wholly. And so my soil samples, I took them last April, around this time of year. The depth increments I took them at were down to 25 centimeters. And the reason for that was just the tillage depth. So to have that treatment comparison. And at the top 2 centimeters, the thing would be different at this is like there's a finer resolution to look at the top 2, looking at the top 2 inches separately, and then looking at every other inch after that down to 10 inches or 25 centimeters. And so the 10 types of phosphorus forms I'm looking at is outside of soil common agronomic P tests. are also runoff and leaching vulnerable phosphorus forms, as well as biologically associated phosphorus. And in this picture is just a picture of me working on some of the biologically associated phosphorus. And so before I get into telling you about some of the results that I've got here, is the way I quantified these is with a stratification ratio. And what I mean by a stratification ratio is it's a severity number that doesn't have any units, and it's supposed to kind of show you how severely or how much more phosphorus you have at a certain depth at your surface depths, and compare it to the amount of phosphorus in the depths below it. So for all the graphs that I'll show you is based off of that equation where the top 5 centimeters were averaged, and that was divided by the average of all the concentrations for the depths below it. And so for this graph, for this slide, I'm showing you the corn and soybean stratification ratios for Malic 3 phosphorus. And what you'll notice is, in general, no-till without a cover crop on average produced a higher stratification ratio, although not significantly. And the same can be said for total phosphorus. Like, again, I'll mention that they have the same equation, is on average, no-till with a winter fallow also was producing the high stratification ratios. However, in the total P with soybeans, there was an exception with chisel plow with a cover crop, and I'm not entirely sure what happened with that there. I'd have to look into that a little bit deeper. But one thing that makes stratification ratios kind of difficult is like comparing between different soil tests. It's like the axis can be much larger or much smaller depending on what it is, because if you have much higher values in your upper soil, like say for water extractable P or soil Malic 3 or soil test phosphorus, you can get much higher stratification ratios, and that's just a reflectable of how stratifiable it is or how vulnerable the stratification it is. And so some of the final remarks I want to make is, in general, no tillage is producing higher average stratification ratios, although not statistically significantly. Cover crops is kind of an interesting addition to this. A lot of studies don't include cover crops, at least in long-term studies, but the effect is generally variable and inconsistent. And some of the effects of other p-tests and pools, which in this presentation, I only showed some of the more common agronomic p-tests, will be shown later on. And in addition, the plant effects of P stratification will be shown later as well, although I also took plant samples during last summer.
Thank you, Reid. Questions from the audience? Okay, I'm going to ask a question that goes back to one of your first slides, the one that showed the figures from Voss and Robinson Voss. Could we look at that again? Is it possible to?
Yes, we can.
Okay. Yeah. So this is for phosphorus, right?
Yes.
And those numbers that I see in the flat no-till, what were the units on those numbers? Do you know? Do you happen to remember?
Milligrams per kilogram.
So near the surface under the flat no-till, the concentration was up near 120 milligrams per kilogram.
Mm-hmm.
And then within 6 inches or so, or no, 10 inches, I guess. No, those are centimeters, 10 centimeters, it dropped down to 40. Mm-hmm. Is that correct? Is that okay? Well, that was my question. I just wanted to review those numbers again, because 120 sounds like a lot. But if my normal soil sampling is to mix all those things together, I don't know whether it's 120 or not in the samples that I take, because I'm mixing them all up. Mm-hmm. Okay, that answers my question.
Have you done already any assessment of risk of loss? Because there's the intersecting factors of the concentration on the no-till or with the cover crop, but potentially decrease in erosion loss. Maybe looking at risk.
question.
Yeah, the question was, she was curious about, am I gonna do any estimation or look at potential losses of, yeah, erosion loss, not for erosion loss or total soil losses. However, with some of my water or some of my P tests, I have estimated some potential dissolved runoff losses. There was a paper done by Antonio Mallarino in about 2006 where they directly measured phosphorus concentrations in runoff and compared it to soil tests at certain depths. And I was able to produce estimates based off of those. And they were all the treatments were close to the same.
Dr. Helmer's online is wondering, what other P fractions are you analyzing for?
Glad you asked. So I mentioned looking at agronomic P tests. So agronomic P tests, I'm showing Malic 3, Olson, and total phosphorus. We have some bicarbonate and calcareous soils on the site, so Olson was another addition to that. The water extractable, water vulnerable phosphorus forms that I'm looking at was water P, so that's just shaking soil and water and then doing colorimetric methods, and that's a good proxy for what could be lost in runoff. And the other one I'm doing that's a little more interesting was with anion exchange resins. So typically when people use anion exchange resins is they'll leave it in the soil for a certain amount of time and then they'll come back to the field and pull it out and then desorb the phosphorus and look at the concentrations. Then I cut mine out and then I mixed it in a centrifuge tube with DI water and then pulled off sorbable phosphorus that way, and then measured it colorimetrically. And then for biological phosphorus, what I'm looking at is microbial biomass phosphorus. And in one of the latter slides, I guess at this point, showed you the desiccation chambers where I was fumigating phosphorus with chloroform gas. And I'm also looking at acid and base phosphatase.
Are there other questions online? Okay, well, thank you very much, Reid. And now I have the pleasure to introduce our third speaker, who is Max Eness. You might have noticed consistency among the speakers so far. The one consistency is they are working with Marshall McDaniel. In the case of Max, he's working with Marshall McDaniel's just since last year. Max grew up in Ames, and he obtained a bachelor's in environmental science at Luther College, which is in Decorah, Iowa, way up in the northeast part of Iowa. And then he began working on his master's degree last fall. And today, this topic is intensive root and soil sampling of perennial ground cover systems. What can we learn? What can we learn?
Thank you, Dr. Thompson, for the introduction. As he said, I'm Maxwell Eness. Today, I'm going to be talking to you about a sampling that we have planned for this June within the Perennial Ground Cover Project. I'm going to be talking about what we hope to learn from looking at the spatial distribution of corn and ground cover roots. And so that's kind of the question that's going to drive this presentation is what can we learn just from looking at where roots are. So to start us off, I kind of want to talk about three dichotomies that are kind of nested and circular in order to kind of explain what we're looking at when we're looking at roots and competition. So the first dichotomy that's kind of the most basic is that when roots interact, they can either be competitive or facilitated. So over on the left here, we have the corn and wheat system. This is a very classic, widely studied intercropping system in which we see yield reductions in both crops. That's not to say that it's not more efficient with nutrients or land use, but both crops perform worse than they would if they were simply monocrops. In the soy corn system, we see an increase in corn yield. This is due to a kind of a synergistic effect. There's a nitrogen credit you get from planting along soy. There's thought to be nitrogen flowing from the soy into the maize, and the soy is not as fiercely competitive for nitrogen as the maize is. So we see a yield increase in maize. The second dichotomy exists kind of within the competition branch of the first dichotomy. There's scramble competition and there is contest competition. So scramble competition, we can see a little facet of it here where the corn is intruding on the soybeans kind of natural territory, how the soy would grow in a monocrop. And they compete over nutrients that are limited. The second branch of this dichotomy is the contest competition. We can see here there's secondary metabolites. A lot of times they're just called secondary compounds. Plants, depending on what species they sense around them, can produce compounds that reduce the competitive plant's ability to grow into a certain location or discourage growth, effectively denying a zone of nutrients for consumption by both plants. So if there's mutual competition going on, a lot of times there's aspects of the soil that are just simply not colonized. The third dichotomy is the symmetrical versus asymmetrical competition. We can see this illustrated in the corn and soy roots relatively well. Because you have an asymmetrical competition occurring here due to the fact that corn shoots out lateral roots very aggressively and is able to reduce the yield of soybeans. And so we see a corn yield boost, but the soy yield decreases, creating that asymmetry. The wheat and corn competition is relatively symmetrical. We see a kind of an equal decrease in yields in those because they're enacting similar pressures on each other. So when you think about those three dichotomies, what our research is going to do is try and explain how those three dichotomies can explain competition between a perennial ground cover and maize. So the Perennial Ground Cover Project is looking at the production of cash crops in between strips of perennial ground cover like you might find in your lawn. Specifically this summer, we're going to be looking at Kentucky bluegrass, which is probably the most dominant lawn grass in Iowa, the Midwest, and Poa bulbosa, which is a European grass that's not widely used for turfs in the Midwest, but it does have pretty strong summer dormancy, which makes it a strong candidate for planting alongside maize. So we reduce competition there. We have observed that there is stress that seems to be induced by cash crops or in cash crops by the presence of PGC, leading to yield drag. Over here, we kind of see a natural ecosystem, and there's, the red lines are supposed to mimic negative or model negative interactions. In the perennial ground system, we're seeing a lot more green lines. Those are positive interactions, but there still exists the possibility of negative interactions, both above ground and below ground in the perennial ground cover system. That's why we're interested in the dichotomies of these competition, so we can begin to understand how the roots of maize are competing with the Kentucky bluegrass and Poa bulbosa. How we plan to do this is we want to construct 2D maps of the roots with both maize planted or maize roots identified and the perennial ground cover roots identified. And Dr. Cole Dutter alongside me will be constructing a 2D map that's at the same scale, looking at nutrients and water, things that they could be competing over. So in order to do this, we've got the 3 treatments that I've already brought up. There's the control, that's monocrop corn, Kentucky bluegrass, and Poa bulbosa. 4 blocks, we're going to be going every 5 centimeters and 9 depths down to 45 centimeters. So we'll have 5 by 5 centimeter samples that we'll be extracting these roots from. We've got boards to ensure that we've got, you know, the proper distance from, or the proper location in the field, and then the proper depth will be done by, you know, just soil knife in field. So these are kind of our sample specifications. We're ultimately collecting 3,600 samples. So it'll be a lot, but half of those will be for root analysis and half of those will be for nutrient analysis. Ultimately, I'll go over a few of the variables that we expect to collect. We're going to be collecting SPAD, which is just a general estimator of plant health, to see if that correlates with anything, see if we're seeing a reduction in plant health that corresponds to changes in root distribution. We are going to be collecting root biomass and root species presence absence, which will allow us to construct that map that we had seen on the left with the, but there's going to be more scale there. So we can, you know, just because there's, you know, tiny lateral roots intersecting doesn't mean that there's necessarily a whole lot of competition. We see a lot of root biomass that could be indicative of, it adds richness to the story. We're also going to, we're going to be using a few methods that might reveal some other interesting chemical changes. We're going to be looking at 13C carbon isotope analysis and near infrared spectrometry, which can provide a, you know, insights into how the chemistry of the roots are changing while not necessarily being, you know, super expensive chemical analyses. So ultimately, going back to the final product we hope to construct, we're hoping to see something, we can kind of see here, the corn roots are not intersecting with these modeled soy roots, where here they are. And then obviously in the, you know, where soy is more intensively planted, you have a lot more intersection. Hopefully the combination of root biomass and presence absence, as well as a control, can show us if the root distribution is changing in corn significantly when we plant perennial ground cover next to it. And so this is an observational study. We are in the one treatment we are really observing is what happens when we introduce maize to perennial ground cover roots. And so I'm going to kind of go through the 3 dichotomies and see, okay, like how might we say that this is the more dominant end of this particular dichotomy when we look at the roots. So there's facilitation. We might be able to see that there's greater plant health in maize when it grows, you know, kind of into the territory of the ground cover. If we see something like that, we might be able to say, hey, there might be some level of facilitation going on here because the corn itself seems to be healthier in the presence of perennial ground cover. Looking at the scramble versus contest, with Cole Dutter's information paired with mine, if we see nutrient reduction in areas in between Poa bulbosa and maize that aren't present in simply the maize, that might indicate that there's scramble competition going on. Likewise, if we see that there's a kind of dead zone between the perennial ground cover and the maize that was not present when the maize was grown as a monoculture, that might be indicative of some allelopathy, some chemical warfare going on between the two systems. Finally, the symmetry and asymmetry, we might see an indication of asymmetry if we see, and this is the hardest one because we're not growing the perennial grasses without corn, so it'll be hard to see who is more harmed by that. But if corn is, you know, it seems to be disrupting root growth or having its root growth disrupted by perennial ground covers, that might be indicative of an asymmetric relationship in favor of the perennial ground covers. Ultimately, this is an observational study, but hopefully it will clarify a few of the further progress that needs to be made with kind of finer tuned competition experiments. You can examine nutrient competition using isotope fertilizers or whatnot, but just getting a picture of the root distribution can give us a lot of really good leads in how the roots are competing. That's all I have. Thank you.
Okay. I think there's time for a couple of questions from the floor here in person. I have one to ask about the difference between the competition dichotomy and the symmetry dichotomy. Now, and maybe I'm oversimplifying this, but it sounded like you were saying the competition dichotomy had to do with competition for nutrients between the two species. Is that correct?
So, so the kind of modes of competition, scramble versus contest. Scramble has a lot more to do with the kind of nutrient depletion. Corn is very good at kind of moving into soy's territory and taking nutrients, water, whatnot away from it. Whereas contest is, so it's kind of the mode at which the root distributions are changed, where asymmetry really is more describing who's the winner and who's the loser in the intercropping system.
Okay. Well, again, maybe I'm just being too simplistic about it. The asymmetry, the way you described the asymmetry or symmetry, it sounded like that was more like competition for space. That one species' roots might be limiting the expansion of others just because there's no more room. Or is that mixing up the concepts?
I think that there's a little bit of concept mixing here, because I think in corn and wheat, there's kind of a mutual antagonism where they both go, their roots are in a narrower column, and they go deeper. They tend to go deeper. And so it's a symmetrical relationship where there's still changes in the roots are they're competing, just not for resources and not in the same space. You could also see kind of an asymmetrical root competition relationship where you have, let's say, through contest competition, where let's say you have a variety of corn that produces a secondary compound that really reduces wheat roots, but the same is not true. They might not be competing in the same exact space, but it's still an asymmetrical relationship. Vice versa could be true. You could have, in theory, a maize plant with, a wonderfully bred legume that doesn't have a crazy nutrient load, but drops in a lot of nitrogen into the soil. And so you have a asymmetrical but facilitative relationship.
Okay. Well, thank you. Are there any questions online, Laura? Do we have any questions? Okay. Well, I think we're right up at maybe a little past the hour, so I want to thank Max for his talk. Thank you very much. And I want to thank each of you who have attended, either in person or online. Be sure to mark your calendars for next Wednesday. We'll have a variety of talks. The topics will be kind of similar to what you've heard today. So be thinking of your questions between now and then too, and we'll continue the discussion on these kinds of topics. So we'll see you all next Wednesday.